Welcome to Microbiology Lab. In today's lab session, you'll be learning about simple staining. So the aim of this experiment is to determine the cell shape, size, and arrangement of bacteria. So as the name suggests, simple staining, in this method, only one stain is used. So the usually basic stains are used such as methylene blue, crystal violet, carbonyl fusion, Grams safranin are the most commonly used simple staining. So these basic stains will readily accept hydrogen ions or it donates the hydroxyl ions which provides the stain positive charge. So we know that the bacterial cell wall, cell surface, it is negatively charged. So these positively charged stains will readily adhere to the cell surface and this helps in visualization. So only the arrangement, shape and size of the bacteria can be visualized by this method. So this is the expected result after performing the experiment. So here you can see only the shape of the bacteria such as rod shaped, cockeye shaped, and then here also you can see the rod shaped bacteria. So only the shape, size and arrangement of bacteria can be visualized by this simple staining method. So now you can visualize the video how the simple staining is performed in the lab.